DNA cycle sequencing is a way of replicating and copying DNA in order to identify a species, such as like a species of a certain disease. DNA cycle sequencing is preferred to the Sanger method because the Sanger method requires you to have a much larger template size. So like the size of the DNA strand that you begin with, for Sanger it has to be much larger than for DNA cycle sequencing, when you can begin with a much smaller amount of DNA sequence to start with. So whenever the first step of DNA cycle sequencing, the cycler, the temperature cycler, it's raised to about 94 to 98 degrees Celsius, and that heat allows for the hydrogen bonds to kind of like separate and turn the double helix into single strand, and that's called denaturation. That's the process of the double helix splitting from the heat and becoming single strands. So after now they're single strands, a primer is going to come, and the step is called annealing. So the primer is going to come, and basically a primer is like a short DNA sequence. And so the primer is going to come, and it's going to bind. It's going to form a hydrogen bond. The temperature inside the cycler has been lowered to about like 40 to 65 degrees Celsius. So the hydrogen bond is able to form, and the primer is able to bind to its complementary site on the template. And so after the primers have bound to the template, then what's going to happen is the cycler is going to, once again, the temperature is going to rise, which allows for the enzyme Taq polymerase to come, and Taq polymerase is going to kind of bind onto the primers, and they're going to, after they're bound onto the primers, then they start to copy the whole DNA sequence. And so once the whole DNA sequence is copied and the temperature is lowered a little bit more, so the hydrogen bonds between them can form, and that is able to get a larger, that helps you to be able to identify the species because you now have a much larger sample size, and with that you're able to find certain species and you're able to identify different diseases and pathogens.